Thank you, Philip, for the kind introduction and the invitation to come here today. So the idea was to try to use another tool to understand difference between us and our closest living relatives. And by that, also trying to understand how we come about to be what we are, humans. And because the pluripotent stem cell technology was being discovered and being refined by the time we were thinking about those questions too in the lab, we decided to use the induced pluripotent stem cell technology, which I will explain soon, to, instead of trying to model for disease the way most people were using those cells for, we decided to see if we could model for human evolution or animal evolution. So I'm, as Philip said before, I'm at Salk Institute. I work with Dr. Fred Gage, and this is in La Jolla, California. So how to study human evolution in the dish? Is that even possible to study human evolution in the dish? Something that is so complex and so complicated. Can we reduce it to the level of the cell? Are we intrinsically different from our closest living relatives at the cellular level, autonomously? So we think that we can try to do that, at least. It's a little bit of a risky thing, but maybe let's just give it a try. So we know that chimpanzees and bonobos are our closest living relatives. Oopsie. And by being our closest living relatives, I mean we share more than, or roughly 97% of DNA sequence, coding regions of DNA sequence. The known coding is still being compared as we speak. We need much more sequencing to be able to make those comparisons, but at least at the coding part, the parts that make proteins, that make us function the way we do, we are 97%, give or take plus or minus 1%, extremely similar to them. How close are we from a banana? 40%. So we're really, really close to these individuals, and yet we are so different, or we think we are so different. And the differences are in brain size, language, susceptibility to infections, susceptibility to neurological diseases. Chimps get Alzheimer's in way less ranges than human. That's questionable too, because the way to address Alzheimer's for a chimp is different than for humans. Cancer susceptibility is different, and also developmental timing is different. We think that if we can identify cellular and molecular difference between humans and non-human primates, specifically chimpanzees and bonobos for that study, we can increase our basic comprehension of evolution and maybe even diversity of our own species in turn. So up until the induced pluripotent stem cell technology was available, the ways we were comparing, researchers were studying evolution, was just by looking at the DNA and getting information by looking at the genetic code. So for instance, I have this gene and chimps don't have it. What would be the meaning of that? But you don't have many models to test, but you can have an idea of existing difference if you look at the genetic material. Another way to study evolution was to look at postmortem tissue. So a brain from a human versus a brain from a chimp, clear difference there. We have more developed Broca area, which is related to language. We have a difference in density of neurons at some areas. But then again, postmortem brain tissues are first very hard to obtain, especially for chimps. Second, so you don't have that much data to support your conclusions. Second, postmortem brain tissues are cells are dead tissues, so they have dead cells. So you lose a lot of the functional information when you look at postmortem brain tissues. You can also look at skeletons. And that can give you some insights along with archaeological evidence about culture, environment, morphology. You can look at endocast, and all those different tools can help you understand evolution in different levels. So as I mentioned before, DNA for genes, tissues, organism, and the culture of the species. However, we believe that if we bring live cells as part of the model, we can improve our understanding, because live cells, studying live neurons from humans and chimps in culture, can help you to make hypotheses and validate those hypotheses looking at a functional neuron in vitro. So to sum up what we did, we got skin samples. So that's the induced pluripotent stem cell technology. You get a cell that was not pluripotent to begin with, such as your skin cell, and you trick these cells by expressing 4 factors, usually, into a pluripotent state. And that technology was developed in 2006, 2007 by a Japanese researcher called Shinya Yamanaka that got the Nobel Prize in 2012 for his discovery. And basically, it makes it possible for you to look at cells that you don't know the DNA exactly, but you can be sure that the DNA is going to be preserved because you differentiate or you reprogram, you change the program of the skin cell into pluripotent cell. So you maintain whatever genetic information was there to begin with, you keep that genetic information. So for the first time, this technology allowed us to look at human neurons, live human neurons, functional in the dish. We were never that able to look at that many numbers before this technology was available, because up until this technology was available, we were only looking at human neurons developed from embryonic stem cells, human embryonic stem cells, and we didn't have that many cells to have an idea of the variation between individuals. So right now, we can have as many induced pluripotent stem cells as we want from as many individuals as we want, and we can compare with chimpanzees, and that it's, for instance, in our case, and that makes it much easier to get this type of tissue than the brain tissue to begin with. So what we do is we get skin cells from humans as well as non-human primates, and I'm going to focus a little bit more on bonobo and chimpanzees because they are the closest relatives to us. And we change the program of these fibroblasts, which are the skin cells, into pluripotent stem cells by adding the 4 factors that have been discovered by Yamanaka. And that's actually a simple procedure. It's just time-consuming, but it's very simple. A lot of people can do, and that's probably why he got the Nobel Prize to begin with, because it's a fundamental discovery that it's not as difficult to do. So once we have the induced pluripotent stem cells, because we induced the fibroblasts to become pluripotent, that's why they're called IPS, we differentiate them into the cell of interest, because we're neuroscientists, we want to look at brain cells or neurons, and we compare neurons from humans versus neurons from non-human primates or chimps in that case. So the idea was to compare those 2 cell populations and ask the question, are there any difference that we can detect at the cellular level that will be relevant to understand human origins or humanness or chimpness? And because it could be that the fact that we look at those cells in vitro would erase any possibility of seeing the difference. And it could be that what makes us human and what makes chimps is just the environment. It has nothing to do with the cells. We wanted to test that. So before I go there, just a quick explanation on what are the stem cells, the cells that we're dealing with. So the stem cells, in that specific case, the induced pluripotent stem cells, are a special type of cells that have 2 main properties: self-renewal, they're able to make more of themselves, and the differentiation is potentially able to differentiate into any other cell from your body. So that's how they look like. They are very round and very compact, and they replicate very fast. And they can make cells from every part of your body. And what we did first was to test our induced pluripotent stem cells from chimps to see if they were really pluripotent. And how do we do that? How do we characterize iPS cells? We look at karyotype. So humans would usually have 46, 2n equals 46, and chimpanzees have 2n equals 48. From chimpanzees to humans, there was a fusion between 2 chromosomes. So humans have less chromosome, even though the genetic material itself is very similar. We also look for expression of pluripotency markers that would tell us if these cells are actually pluripotent. And we look at NANOG and TRAL181. Those are the two main markers. And we also look at general differentiation. For instance, how able is the stem cell to make bodies that have, to make cell cultures that have the 3 embryonic layers: ectoderm, endoderm, mesoderm. That will also tell us this is a pluripotent stem cell. It can make the 3 embryonic layers of an individual. So for that, we make embryoid bodies in vivo. Oh, that's just the characterization of the iPS stem cells. Those are the karyotypes. You can't really see so well, but trust me, they were stable at that point. We did 2 chimpanzees, 2 bonobos, and 4 humans. And the human we did was not me. We're not allowed to culture our own cells in the lab. Just because we break the barrier of infection. So we tend not to culture our own cells. So what we did is we differentiate first in vitro to show that the bodies that we can make by adding specific factors will contain the 3 embryonic layers, which are endoderm, mesoderm, and ectoderm, here characterized by the 3 different factors: Musashi, Brachy, and AFP. And we also show that those cells can form teratomas in vivo. So if you put them if you inject those cells into a nude mice, we can see the same structure forming. We are not making any clones, but we just have an idea that those cells can differentiate. So then what we did, we wanted to know if those cells were differentiating to neurostem cells or neuroprogenitor cells, which are the cells that are going to give rise to more differentiated neurons or brain cells. And in order to do that, we plated the embryoid bodies under neuralizing conditions, and we looked at neurorosettes. And neurorosettes are primordions of the neural tube. And then we dissociated those neurorosettes into a monolayer of cells that we call neuroprogenitor cells. Those cells can give rise to neurons if you further differentiate them and wait in the culture. And that's how the neurorosettes look in the culture. And that's how the NPCs look in the culture. They're very flat, and they have this fusiform appearance. Well, we looked at if we kept them further along in the culture, would they differentiate into neurons? And we can obtain neurons marked here by a neuronal marker, specific poneuronal marker called TUG1. And the cells, chimpanzees, and both chimpanzees and bonobo cells can make neurons in culture. So those are all good news. And it meant to us that they were mirroring what was happening to human cells. So where would we expect difference at that time? So we're trying to think which experiments to do with those cells in culture that could give us some insights that could tease the difference between humans and chimps. So, and just out of general knowledge, the difference that you would expect are in the regulation of gene expression. So some genes will be starting their expression profile earlier and some would start later. So there is a timing difference, not necessarily in neurons and genes that we have and chimps don't have, but in the timing of expression. And we also know that brain size and brain organization in humans and chimps differ. We know that human brain is about 3 pounds, and chimp brain is roughly three times smaller. Even though the skull sizes are somewhat comparable, the actual brain size that would go into the cranium is much smaller in chimps. So we first tried an unbiased approach. We looked at, we performed high-throughput RNA sequencing in the neuroprogenitor cells to look at the differential genes that were, or groups of genes that were expressed in human cells versus chimpanzees and bonobos. And just a quick recap on what is RNA, how do we prepare RNA-seq library. We basically purify our RNA. We bind poly-A, so then we know that those RNAs are expressed. Then we fragment our RNA, and we convert to cDNA. And following the conversion, we apply adapters, and we sequence all mRNA from humans and non-human primates. And when we did that, compared to non-human primates, we detected approximately 2,000 genes that were differentially regulated. But interestingly, within the 2,500 genes, we found changes in categories that are related to cell migration, according to gene ontology. So we had our first hint that maybe if we look at migration, we might be able to see some measurable difference. That's what we did. We set up a simple migration assay called the scratch assay. So we basically scratch the surface of a plate using a glass pipette, and then we score individual cells migrating towards the center of the scratch. That's just how they look at the bright field. So the cells tend to migrate towards the scratch. So we also quantified is the directional migration. The cells are migrating. They're not migrating randomly. They're migrating towards the scratch. And then we marked individual cells and follow their path, so then we could get insights of direction and speed of migration. And when we do that over time, we noticed that after 24 hours, most of the chimp lines had completely closed the gap, while the human lines haven't closed the gap yet. So that was interesting and promising, but we wanted to have a more unbiased way. We wanted to see if we could look at both cells at the same plate and see the difference, instead of looking at one plate for humans and one plate of the chimps. So what we did is we took advantage of microfluidic chamber assays. We have a bioengineer guy in the lab, and he designed microfluidic chambers where we could have just culture media in one side and human mixed with chimp neuroprogenitor cells at the other side. And what we did to know the difference between one and the other, we color-coded them. We expressed a green fluorescent protein in one and a red fluorescent protein in the other, so we could find out who was who. And also, those microgrooves were just small enough for one cell to go at a time. So we set up that experiment, and we just observed what was happening. So that's basically the cells on one side, the green and red mixed, and after 5 days, most of them are migrating. And we recorded the migration so we could quantify the speed. So that's shown here. Those are the microgrooves. I like to put that picture so you can see how thin is the microgroove and the cell kind of squeeze within it. And I would like to show you the other video without only at the fluorescence. And I can, based on what you've seen before, can you tell me which is which? If green, which is human and which is chimp? Chimp is red. Yeah, chimp is red. So we were happy to use we used different experiments that were showing similar things. So that's always good for scientists when you see similar things in different experiments set up. So we then went ahead and quantified. So we quantified all the cells in the grooves, and we quantified all the cells migrating towards the scratch. And indeed, we detect decreased speed in human NPCs, neuroprogenitor cells, compared to chimpanzees and bonobos. So both chimpanzees and bonobos were migrating faster. We are still investigating what are the reasons for it. The number of genes that are differentially regulated is big. So we try to do an approach which we would knock out gene by gene, but there is not one gene that is responsible for the changes. It's probably a combination of things, of genes. So we're still investigating what are the causes for the difference, but the difference seems to be consistent. Humans are slower than chimps. We have seen difference in differentiation, but we weren't able to reproduce to see any significant difference at the neuronal differentiation. That's what we are. So can we detect difference between human and non-human primate neurons? And when we did that in vitro, if you look at the bottom panel, it's the quantifications for both markers for neuronal for more mature neuronal differentiations. One is MAP2, the other one is synapsin. And we do not see obvious difference. So that made us wonder if we were looking at the not so stringent system to detect the difference, because we're just looking at in vitro neurons. And even though we're looking at function right now, we had the idea that we could be masking some of the difference because we are treating everybody so similar, and the in vitro pressure can be so heavy. So what we did is we took advantage of a natural 3D system, which is the mouse brain, and we transplanted those cells into the mouse brain, and taking advantage that there is a 3D system in place that can allow for the neurons to thrive more in terms of 3D differentiation. So we know that the development of human cortical neurons follow specific developmental hallmarks, and we know that chimps follow a specific developmental hallmark too. However, we never saw them how they behave together. So what we set up to do is we transplanted human neuroprogenitor cells and chimp neuroprogenitor cells into mice brains, and we look at how those cells were maturing over time. And we looked at parameters that are total dendritic length, total dendritic segment, mean segment, dendritic spine number, dendritic spine density, number of densities, and cell body. Those are morphological measurements for the type of neurons that we were looking at. We only looked at cortical neurons that we're not necessarily able to make them in vitro. So we performed stereotaxic injections, and we went on into look for the different morphologies of those neurons. And we used this software to trace the neurons and give us specific measurements of size of the processes, branching, and number of spines. Spines are the protrusions that neurons communicate to the others. That's where the synapse occurs. That's where the communication occurs. So number of spines is it's also a way to look at functionality. So the higher the number of spines, the more mature your neuron is. So when we compare those two populations, and here chimpanzees are in red, pen, and Homo sapiens in green, we notice a clear difference in pyramidal neurons with regards to total dendritic length. So human neurons go further. It takes a while, but then chimpanzees kind of stagger, and then human neurons differentiate further. And we also saw a significant increase in the spine number. As I mentioned before, spine number is the communication between neurons, is a measurement of how integrated these neurons are. And the beauty of that type of research, of that type of experiment, is the brain of the mouse is completely agnostic. The system is completely agnostic towards the cell. So if the difference that we see are cell autonomous to the human neurons and the chimp neurons. So they are the ones that are different. The mouse brain is, the mouse neurons are not supposedly changing their, they're just receiving them, but the difference are specific. So we are actually looking at cell autonomous difference between humans and chimp neurons. So conclusions for that part of the talk will be we can use IPS and neuroprogenitors and neurons, and we can generate them from human primates. Human neuroprogenitors or neuroprogenitor cells can migrate. They do migrate slower than the chimps. Human and chimps IPS-derived neurons can recapitulate developmental hallmarks in vivo, even though they are into the mouse brain. And human pyramidal neurons in the mouse brain show faster maturation at later time points compared to chimpanzee neurons, as measured by increased dendritic length and spine number. And also, the transcription profile of human and non-human primates, neuroprogenitors, and neurons, we're analyzing that now, show differential gene regulation. So we think, and we're proposing here, that we can use primate IPS as a model to study comparative difference between humans and non-human primates. And we think that those differences could have an evolutionary impact. And we are furthering studying how to change, what are the changes involved in that increased differentiation in human neurons at later time points. In future directions, we would like to include other non-human primates to compare to also human, such as gorilla, orangutans, and macaques. We would like to have a better idea of molecular signature during neuronal development. So now we are doing RNA sequencing on cells that have been differentiated in vitro for 2 weeks, 4 weeks, 6 weeks, 8 weeks, 19 weeks. So we can compare the differentiation signature from humans and non-human primates and understand when is the time of change. According to our in vitro studies, we think it's around 2 to 3 weeks, but we like to investigate further. And that could be important because if you hold the differentiation, so then you go slower in the beginning, do you become more like a chimp-like neuron, or you become more like a human-like neuron? So we're trying to find what are the parameters that control the differentiation process. differentially. And we also would like to compare neuronal function and connectivity. This is, as I mentioned before, a unique opportunity to record activity of human live neurons and compare to non-human primate neurons. Up until now, we were not able to record electrical activity from any primate neurons, at least not apes, because we can't have access to their brains. And I would like to thank you all for coming, and just give a quick thanks for Rusty Gage, my mentor and colleague. Inigo, Bilal, Ahmed, Diana, and Krishna were directly involved into that project. And a few collaborators from UCSD, Katerina, Alison, and Jean. And we have funding from Matters Foundation. That's Rusty Gage, the head of the lab. And I'd like to thank you all, and I take questions if you have. Thank you.